Okay, today I want to wrap up our discussion of water balance in plants, focusing today mostly on water movement through the leaf and from the leaf to the air. Here are the learning objectives for today. Please make sure you understand all of these concepts and come and speak with me if you have any questions on any of the content of today's online lecture. We've been discussing how water moves through the plant, and we've been modeling the movement of water through the plant using Ohm's law. So we have been setting up this analogy that water movement in the plant is similar to the movement of electrons, and that it can be calculated, its rate or current can be calculated based on the driving force, which is the difference in water potential between two locations, and the resistance to water movement between those two locations. So we discussed water movement in the soil, and we discussed water movement through the root and the different ways that the root resists, or the inverse of resistance is conductance, or facilitates the movement of water. And then we ended with water movement in the xylem. And what we established is that resistance in the xylem is determined by things such as the width of the xylem cells, and also whether or not the xylem cells are clogged, and those clogs in plants come in the form of embolisms or air bubbles, which can be detrimental to the plant. But plants have evolved mechanisms in order to deal with embolisms, and plants in certain environments, like plants that live under very dry conditions, are really good at dealing with embolisms and preventing embolisms from occurring even under very high tension forces in the xylem. So what I'd like to talk about today is how these negative tension forces form in the leaves, and also how water moves through the leaves and then into the environment. So we're going to be focusing on this aspect of the soy plant atmosphere continuum today. Okay, so we talked about xylem, and in general, there is not a lot of resistance to water movement through the xylem. And we know this because they're basically hollow tubes, right? And the cell walls are made of cellulose, which is hydrophilic, so it has the benefit of capillarity. And there's no cell membranes to cross, and as long as there's no bubbles in it, resistance to the xylem is very low. And so you only need a pretty small pressure difference in order to get water to move in the xylem. And the reason is because of Ohm's law, I equals V over R. So if R is small, the driving force, or the difference in water potential, can also be small in order for you to get a decent rate of flow. So scientists have gone and calculated how large of a pressure difference would be required to move water through the xylem from even the tallest trees, so the coastal redwoods, which can be over 100 meters tall, almost 120 meters tall. They have to factor in gravity potential because of the height difference of these trees, but even when you factor that in, you only need a pressure difference of about 2 megapascals from the top of the tree to the bottom of the tree. So it really doesn't require all that much of a difference in pressure. So let's talk about how this pressure gradient is established. So in theory, there's two ways that you could have a pressure gradient, right? You could have positive pressure at the bottom, pushing water up to a lower potential at the top of the tree. Or you could remember that water can have negative pressure, or we could have a really, really, really low negative tension at the top of the tree that pulls the water up from the base of the tree. So we know that when there is positive pressure occurring, so again, that can happen in the morning after the stomata close, when you have all those solutes that build up in the xylem and water is absorbed, and you have this positive pressure in the bottom of the xylem, it's only about 0.1 megapascals. And this isn't going to provide the needed pressure differential in order to get water to move up through the xylem. This pressure is also alleviated once the stomata open and water begins to evaporate. So the way that water moves from the base of the tree to the top of the tree is because of the very, very high negative tension force that develops in the leaves. And this is going to provide the driving force needed to move water from a higher water potential in the roots at the base of the xylem to a lower negative potential, and that is at the tops of the trees. This mechanism, of course, is going to rely on those cool properties of water molecules that occur because of the intermolecular interactions that form from hydrogen bonding. So capillarity and cohesion and having a very high tensile strength. This is all going to be crucial for this cohesion tension theory to happen. And so the negative tension that forms at the top of the leaf is caused by the evaporation of water from the stomata. So it's going to require an input of energy, and that energy comes from the sun. And this is going to create the water potential gradient going from the leaf air spaces that have a very, very low water potential to the base of the tree, which will be much higher. So let's talk about how that negative tension forms in the leaves. So that negative tension is actually going to form in the mesophyll. So if you remember what the leaf looks like, you have the epidermal cells, the top and the bottom. So you have the adaxial and the abaxial epidermis. They should have a guard cell here for accuracy's sake, but they don't. And then you have this palisade parenchyma here, which are going to be the photosynthetic cells. And then you have the spongy mesophyll cells here. And then there's all these air spaces around them. So, and remember that the cell walls here are made up of cellulose, which is incredibly hydrophilic. So it's going to have a lot of adhesion. It's going to want to be in contact with water. So as the water gets evaporated, so as the water on the surface of these cells moves from liquid water to vapor, what's going to happen is the water that's going to get evaporated first is going to be where there's more water, okay? And so that water is going to get evaporated, and what that means is the remaining water is going to cling even more tightly to these small spaces between the cellulose microfibrils that are going to adhere to the water. And what this does is just like what happened in the soil, it's going to create more curved air-water interfaces. And that is going to be the source of the negative tension in the leaves. So remember that the air and water, they want to minimize their interaction. Water molecules don't want to interact with air. They would rather interact with the cellulose microfibrils of the cell walls and of the other water molecules. So because they're being forced to interact more with air, this is where that negative tension force that's going to power the movement of water from the base of the tree to the leaves gets generated. And of course, the driving force for that will be provided by the sun. That energy needed will be from the sun as it evaporates water by warming the leaf. Now, the tree also has even though the tree's not actively investing in the movement of water, that it's coming from the sun, the tree still has to invest in this process because there's going to be really, really, really negative tension forces in the xylem, especially in those very tall trees. And that column of water has to maintain its tensile strength. If that column of water breaks, the whole mechanism fails. So what the tree has to do, or the plant has to do, is it has to invest into the cell walls of the xylem by making them really, really strong. And that's why lignin and cellulose are so important. And it has to continue to make more and more xylem to support that column of water in the tree. And also these mesophyll cells, they have to maintain turgor even though like they have this extremely high tension force. So that means they're going to have to be constantly accumulating solutes to maintain turgor pressure when exposed to these really, really low pressure potentials. Okay, so now let's talk about water movement from the xylem to the leaves to the atmosphere. So in the leaves, there's going to be 3 elements to the water flow: through the veins, from the veins to the stomata, and out through the stomata. And the flow through each of these pathways is going to be determined by the resistances in each of these pathways, right? So we'll have a resistance from the vascular system. Once it hits the cells, then it can move through different pathways. It can move through the apoplasm, or it can move through the symplasm. It can move cell to cell or the transmembrane pathway. It can also switch between pathways, so it can move, the water can move through the apoplasm and then enter the symplasm and then go out through the stomatopore. Okay, we can also talk about leaf conductance in terms of the xylem conductance and then the out of xylem conductance. So here, conductance I'm referring to as K leaf, and xylem conductance would be Kx, and out of xylem conductance is Kox. So Kx is going to be determined by things that we talked about last time. So the xylem conduit diameter, is it clogged? But also things like the anatomy and the abundance of veins in the leaves. Not all leaves have the same number of veins, and if you have more veins, you're going to be able to transmit water more efficiently. And out of xylem conductance is going to be determined by the non-vascular cells, right? So things like how far is the distance between the veins and the stomata? What's the density of stomata? How many portals are there for the water to leave the cell? And also how open and closed are the stomata? So these are some of the things we're going to look at today. So first is xylem anatomy. So the xylem in the leaf is very efficient, and it's going to be a low resistance pathway to water movement. And in most leaves, the vein pattern is such that the photosynthetic cells are located all very close to a vein. And this is going to ensure a steady supply of water and a relatively short pathway to return the products of photosynthesis to the vascular tissue. And what we see is we see differences in vein density between angiosperms and non-angiosperms. And we see that the vein density of angiosperms is quite high compared to non-angiosperms. And that high density may be essential to support the high rates of transpiration and photosynthesis, which we see in angiosperms, right? So non-angiosperms aren't very tall, right? Some of them don't even have vascular tissue at all. So the ones that do have vascular tissue, like ferns and gymnosperms, gymnosperms are able to get tall, of course, but we see more vein density in angiosperms, and we think that this is part of the reason angiosperms have been so successful is because they're able to maintain higher rates of transpiration, and they're able to photosynthesize more than some of the other plant groups. It also provides kind of like a backup mechanism, right? So if you have more veins, if one of them gets clogged, then you will have an alternative path and you won't completely clog up the water pathway. Okay, I also want to mention another type of cells. We talked about these in 240. They're called the bundle sheath cells, okay? So in angiosperms, the leaf vasculature is surrounded by specialized mesophyll cells. These are called the bundle sheath cells. They also have an important role in C4 photosynthesis that we talked about in 240, and we'll be talking about that again later in the semester. But as water leaves the xylem, they're actually going to first pass through these bundle sheath cells So here's the xylem here. This is the xylem tissue, and then these are the bundle sheath cells here. So they are going to be in between the veins and the mesophyll cells of the leaf. I mentioned before that the mesophyll cells have to maintain a very low osmotic potential in order to prevent the backward flow of water back into these mesophyll cells and into the veins. Okay? So water leaving the xylem passes through the bundle sheath cells, and we think that they can vary in conductance with different environmental conditions. So they can kind of serve as a gatekeeper to how much water is leaving the cell. And there's also some evidence to suggest bundle sheath cells have a similar role to the endodermis. Remember, the endodermis acted like a filter, and it could control the flow of solutes into the leaf tissue as well. So the bundle sheath cells kind of have a regulatory role in the leaves. And the hydraulic conductance of the leaves from the vein to the somatopore are partially regulated also by ABA and by drought signals. And they are also likely to be mediated, at least in part, by aquaporin activities. Remember, aquaporins are those water channels that exist in cells. There's quite a few of them in plants. And so the position and the abundance and the status of these aquaporin channels are also going to help regulate water movement between the veins and into the leaf spaces. Okay? And again, this is going to regulate the out of xylem pathway in the leaf. Okay, let's talk a little bit more about what happens in the leaf, right? So the negative tension forces we established are going to occur here, right, in the mesophyll cells. That's going to drive water movement here into the mesophyll cells. And there's a lot of surface area in there, right? So there's this spongy mesophyll. They're not jam-packed. There's a lot of room for interaction between the cells and the air spaces. And so as water moves into these mesophyll cells, the water is going to get warmed up because of the sun is going to hit the leaf. It's going to warm it. And what's going to happen is that water clinging on the surface of these mesophyll cells on the outside is going to get evaporated, okay? And the air spaces here, because of all this water moving in the plant, moving into the mesophyll, it's going to be nearly at saturation, okay? So it's going to be incredibly humid here in these air spaces. And we can actually estimate the concentration of water in the leaf, these air spaces, based on the leaf temperature. Because saturated water volume content of the air actually increases exponentially with temperature. And that's because warm air can hold more water than dry air. So this is an important relationship. So as the leaf temperature, we can measure the leaf temperature and we can calculate the amount of water or the vapor pressure of water in that leaf space in the air. And we're going to be using this idea later on in the semester when we talk about gas exchange measurements. Okay? All right, so what we have here is we have really, really super moist, high concentration of air in these air spaces. This is important to note. The final stage of water movement, so water movement from the leaf to the atmosphere, is controlled by the concentration gradient of water. Okay? There's more water vapor within the leaf in those air spaces than in the atmosphere. And the concentration of water in the leaf depends, remember, on the temperature of the leaf. Okay? So water at this point is going to move from diffusion down a concentration gradient. So here we're showing the relative humidity. So you can see it's about 99% in the air spaces of the leaf. And at this temperature, we can calculate what that concentration is. And it's about 1.7 moles per per meter cubed, okay? And then just outside the stomatopore, we see a much lower relative humidity, and we can calculate the concentration at 0.6. And then bulk air at 20 degrees, we have a concentration of 0.50. Okay, even though it's more humid out here, the actual concentration is going to decrease because the temperature is lower and it can't hold as much water vapor. So it's this concentration gradient that is going to be the driving force for water movement from the leaf spaces to the atmosphere. Oops. I don't know why that's cut off. And an important component in controlling the diffusion of water out from the air spaces to the leaves is going to be the guard cells. Okay, so guard cells are the gatekeepers of transpiration. They are going to induce a stomatal resistance to the evaporation and the diffusion of water out of the leaf. Okay. So an analogy to this is it doesn't matter how quickly you can jump out of your airplane seat until the door opens, you're not going anywhere, right? So even if there's this huge concentration gradient here, if the guard cells have a very high resistance, if their pores are not open, then water's not going to be able to leave the plant. So closed stomata, we're going to have no transpiration and no flow. Open stomata, we're going to have low resistance, high conductance, and we're going to have flow that could potentially be regulated by other factors. But these stomata are going to be the driving or the main source of resistance to water flow from the air spaces to the atmosphere. So how do they do this? So guard cells are going to control the pore size that CO2 enters and exits the leaf, right? So CO2 will enter the leaf through this pore, water will diffuse out of the plant through this pore. And guard cells are able to integrate all sorts of environmental signals to figure out the optimal stomatal aperture size. They respond to ABA, which is the stress hormone. They respond to CO2 concentration. They respond to water potential differences in the roots. They respond to light. They can respond to all sorts of different things. They're pretty amazing in how they integrate signals into a particular response. And I want to talk a little bit more about the dynamics and the anatomy, how the anatomy allows these cells to do that. Oh, I thought I was going to talk about something else first. Okay, so what I have here are some images of the anatomy of the guard cell. So there's generally two kinds of stomata you could have, okay? These stomata here are seen, these are kidney-shaped guard cells. And you have two kidney-shaped guard cells, and they're flanking a pore here. And they're surrounded by epidermal cells. We see this a lot in most eudicots. This is the other kind. We call these like the dumbbell-shaped guard cells. They work a little different. They're kind of like beams that have inflatable ends. So what happens is when these cells gain water, the ends inflate and they open the slit. And when they lose water, they collapse and they close the slit. So this is an example of guard cells in the monocot. You can see that they have these really thick, differentially thick cell walls here. So they're thick on this side and then they're thinner in the other portions. So water expansion is going to occur in this portion of the cell, not so much in this portion of the cell because the cell wall is so thick. Okay, so these are going to expand. There's not going to be an expansion here. These cells are going to push on each other and the slit's going to open. The guard cells here, this is what a kidney-shaped cell looks like in cross-section. They also have like a differential thickness in the cell wall. So, and they also have these radially arranged cellulose microfibrils in the cell wall. And this is really important because this cell wall here is thicker than the outer cell wall. And also, because these are arranged radially, it's much stronger and less flexible. So when these cells fill with water, this part, the outer part, is going to expand more than the inner part, and the pore is going to open. Okay? So the structure of these cells is critical to how they function. I also want to note these are called subsidiary cells, and they're a type of epidermal cell that is in next to a guard cell, and they are able to assist with the opening and closing of stomatal pores. So what we'll see is that these changes in turgor in the guard cells has to do with the flow of ions and the flow of water between in and to and out of the cell. These cells act as reservoirs for that, but they also can, when ions and water leave the cells, they go into the subsidiary cells, which then gain water, and then they push on the guard cells and they assist in the closure of the pore. Okay, so it's a really interesting anatomy. So these guard cell movements are controlled by ion currents. So guard cells are able to integrate all those different environmental signals, and they end up activating or inactivating various ion channels, and this causes ions to leave the cell. Some of these ions in water will come from vacuoles, but what happens is when the guard cells so this is an open stomata, and when it's time to close because maybe intracellular CO2 concentration is high or ABA is getting synthesized in the plant, ion channels in the membranes actually will open, releasing ions from the cell. Okay, so these ion channels will release ions, and then what happens is these ions will draw water out of the cell. Water will just follow by osmosis. So the ions leave the cell, and then the water is going to leave the cell. Just like we talked about the first day when we were talking about water movements in the cell. Okay, so the osmotic potential goes down, and the water potential goes up, and then it moves from higher to low. Guard cells lose the turgor pressure and relax, and then they close the pore. Okay, so when the turgor goes down, the pores close. They're no longer pushing on those cell walls, and we have closed stomata. As I mentioned before, many different factors regulate stomata: CO2 concentration, drought, ABA, light. These are all really, really important. They also respond to hydraulic signals and changes in water potential. Stomatal density and aperture size and also the anatomy of the leaf can also affect hydraulic conductance or leaf out of xylem resistance. Some of these things include like stomatal crypts. So the stomatal crypts the stomata are actually located kind of in these invaginations in the leaves, and this increases the humidity outside of the stomata, so it reduces the concentration gradient, so less water diffuses out of the leaf. So this is one type of leaf anatomy that contributes to it. Another has to do with density. So if there's a lot of stomata, there's going to be an increase in stomatal conductance compared to a leaf that has fewer stomata. And also, as I mentioned already, the subsidiary cells can be important players in these movements. They can provide sources of ions, and they can be a source of water too to regulate the movement of stomata. And then in the case of some of the grasses, they actually make it more efficient by pushing the guard cells together or losing water and giving the guard cells room to expand and open the pore. I want to mention boundary layer resistance, which is another aspect of the diffusion, another factor that adds resistance to water movement from the leaf to the atmosphere. And what the boundary layer is, it's a layer of still air, of unmoving air on the surface of the leaf. And this is called the boundary layer. It's thick and it resists the diffusion of water from the leaf to the atmosphere. So what happens when air is moving, say it's windier outside, the boundary layer gets thinner and resistance decreases. So what this is showing here, this is showing the rate of transpiration at a given stomatal aperture with different boundary layer conditions. So the black line here is representing moving air, and the red line is still air. And you can see that the transpiration rate at a given stomatal aperture is much greater when air is moving over the surface of the leaf as opposed to completely still air. So this can be a significant factor in regulating the amount of water that diffuses from the leaf. There are also different aspects of plant anatomy that can contribute to the boundary layer resistance. One is our trichomes. So those are those little leafy appendages, those leaf hairs, and they act as windbreaks, and they can actually increase the boundary layer resistance and reduce the amount of water that's lost from the leaf. Also, things like leaf orientation, wind speed and direction, all of these are going to affect the boundary layer resistance. Okay, that's all I have for today. If you have any questions on anything I covered, please bring them to class or come visit me in office hours. Okay, thank you. Bye.